Good afternoon, everyone. It is my pleasure to introduce Prof Bellstedt, Emeritus Professor of Biochemistry at Stellenbosch University and a leading expert in biochemistry, molecular biology, and immunology. His research spans plant and animal pathogens, vaccine development, phylogenetics, and biochemistry with over 90 publications and collaborations across Africa and Europe. Prof Bellstedt's distinguished career has also includes his pioneering work in diagnostic techniques, such as the development of ELISA tests for bacterial wealth in potatoes, which is still being used and has been adopted nationwide for seed certification. He is a dedicated author, educator, and mentor, supervising more than 60 postgraduate students and chairing the Stellenbosch University Animal Ethics Committee. We are honored to have him here today to share his insights on the applications of harm-benefit analyses in animal research and trial and/or experimental design. Please join me in welcoming Prof Bellstedt.
Thank you, Biosha. I feel very flattered. Thank you. So I see we are a small audience, which is comfortable, and it means that we can discuss this topic today at leisure. I welcome any interjections if you have any, if you would like to ask specific questions. What I actually want to do, and Biosha, you can go to the first slide, please. I actually don't just want to discuss the harm-benefit analysis because I'm a little concerned that that might place it somewhat just out of context. So what I want to do is I want to give you a little bit of background on animal ethics applications and what in general is important, and then highlight a few things about the process, not detail, but just to give you some guidance. And then I want to eventually say that we use assessment tools to assess animal ethics applications. And amongst those, the harm-benefit analysis is one that can often help us in particularly sticky situations. Not always, but it's a tool and it's an interesting one. So if we can go to the next slide, please. The question is, why do we need animal ethics clearance if we do animal experiments at Stellenbosch University? And the first thing is that through national legislation, we are required by the National Department of Health that all animal experiments be ethically approved. So that's a legal obligation that the university is under, and as a result of that, that is one of the reasons why this committee exists. And then there is a very simple one, and that is that in modern day animal research, if you want to publish that work, you will be asked by the journal whether there has been ethics clearance. If you have applied, you will have an ethics clearance number. You can specify that, and that's the end of the story. So this is an important issue for young career scientists to remember that this needs to be in place. And then in any signing of your employment contract, often one doesn't read the fine print of these employment contracts, but there you have already signed that should you do any animal experiments that you would apply for animal ethics clearance. And this is also part of the enrollments of students, and it is required of you. And what is very important is the last part where I've just highlighted that this be done before. We cannot retrospectively give animal ethics clearances. So this has to happen before. But it's not a monster, can actually be done quite easily. So the research involving animals includes medical research, rats, mice, rabbits, etc. But we've even go as far as pigs and other animals which have served as models for humans and which we then use in animal research. Animal husbandry, the university has a department in which animal husbandry is practiced, and trials on animal feeds, etc. are done by them, and they require ethics clearance. And then we have a number of departments, zoology and conservation biology, in which animals are sampled for biodiversity assessments, and a variety of other purposes. So those are the fields that we give ethics clearance in. And now you might ask, well, how does this happen? Are there rules? Yes, there are rules. There are the South African National Standard 10386, which gives you guidelines for how experimental animals should be housed and what they should be fed, etc., etc. And those need to be adhered to. That document is available on the research development's website. So should you wish to fill in an application, you can consult that. And then if you're doing research on pathogens of animals, then you need to apply to the Department of Agriculture separately from our application for a so-called Section 20 permit. And again, the Division of Research Development, specifically the animal ethics staff, can give you guidance and assist you in how to approach such a Section 20 permit. So these are important matters for consideration if you wish to perform animal research. There is an application process which is done online. It's, as far as I'm concerned, a system which really works well. You can go to this link, as I've indicated to you, and you can download you can look at the whole process. It is extensive, and that is because animal research has to be done thoroughly and has to be done correctly. And as a result of that, this is not a process that you can just do late one afternoon. You're going to have to work your way into it. The applications must be submitted by certain deadlines. There are dates as indicated on Research Development's website, and we strive to give outcomes of applications within 6 weeks. If there are problems, we will go and actually try to accommodate the researcher and hold an online session or meet up and try to explain to them what is required if there are things that are unclear. So the whole process is actually a support process for the researcher to enable you to do the research, not to try to administratively hinder you. So next page, please. So an animal ethics application using animals can only be justified if there is a scientifically sound motivation. Then we need to know which animal species. That's completely easy to understand, and we need numbers, and the numbers are critical because we need to assess how much harm is going to be done. And then it may be because of regulations. If you want to draw blood from an animal or something like that, you are not permitted to do that without Veterinary Council approval. The Veterinary Council is the registering body for all vets, but they will also give you special permission by means of accreditation to perform certain procedures. And the vets that the university employs, there is Dr. S.D. Spies in the medical and science faculties, and a vet also in the agriculture facility, Dr. Benny Grobler, and they can assist you and train you so that you can perform those procedures. Once you're competent, you become, you actually receive an accreditation. You are registered with the SAVC, and it allows you to do these procedures. Next page, please. So how does this get assessed? The Research Ethics Committee, Animal Care and Use, consists of scientific experts in all of these fields: medical, agriculture, conservation, biology. I think the team at this stage, I'm not entirely sure, but I think there are 14 scientists on the committee, 2 veterinarians, and then there are a number of lay persons, and those lay persons are required by the National Department of Health. And I actually think that they do a very fine job in that they just simply put this issue right down, plain down to earth, and they say, well, can you justify animal use? And then the division of research development, and this is an important point. We want to assist and enable you to do research, not delay or hinder you with unnecessary administration. And that is my considered opinion, and I push for that. So how do we evaluate applications? We specifically ask, is the use of animals justified? Will the use of animals give answers to research questions? Very often animal experiments are ill-defined, but they need to precisely address the problem, and the research that is performed must be able to give an answer to the questions that are asked. And then, are the numbers justified? We also have a statistician on board, and the numbers are examined with regard to whether the statistical variability is such that with the numbers that you require, either that the numbers be too few, surprisingly enough, because if the numbers are too few and you can't actually you can't prove what you wish, then the few animals that you've used have actually been wasted. So it may be that in actual fact the committee might recommend that you use more animals, or in some instances use less. So there is also a benefit from interaction with the committee in terms of the statistics. But finally, and most importantly, does the benefit of the trial justify the use and the suffering of the animals that are to be used in the trial? And this is critical. We will not allow unnecessary suffering, and we will not allow certain animals to be used. I don't know whether you perhaps have been following all of the turmoil with regard to research funding in the United States, but one of the facilities in the United States that's influenced is a chimpanzee facility. And it's not sure what's going to happen to these poor animals. Stellenbosch University's Ethics Committee does not consider work with primates. We do not wish to use primates because of the sensitivity of these animals. So just to get to the harm-benefit assessment, because this is what it's all about. And the harm-benefit assessment is a checklist that is drawn up of all the factors relevant to an animal ethics application. And we try to do this, although this sounds somewhat bizarre, we try to move away from subjectivity to objectivity. In other words, we don't just say, oh, yes, but you know, those animals are lovely and we don't want them to be harmed, and so on. We say, let's put this all together into some sort of a formula, and this is what the harm-benefit assessment is, which will hopefully give us some sort of weighting system so that we can eventually put it all together and then decide whether the harm outweighs the benefit or the other way around. And we try to assign numerical weights to a variety of factors that have an influence on animal trials. And this now sounds a bit bizarre because these terms are a bit funny, but I'm specifically referring to one of these tools, and that is we calculate the interest of the ultimate goal. In other words, is it justified? We assess the humaneness and the relevance, and we combine both in an assessment of human interest. So this is why are we wanting to do this? We're weighing this up against the animals. So human interest, one back please. Can the findings contribute to prevention of human and animal suffering, morbidity or death, halt or reverse ecological and environmental harm? In this instance, in this explanation here, I've expanded on this to not only include human suffering. I mean, a lot of animal trials are done so as to study cancer. And cancer is a huge problem in humans and causes incredible suffering. And so we need to weigh up whether the animal suffering can be such that we can live with that relative to the human suffering. And you can see these are factors which are difficult to weigh up. And then one of the modern things that's creeping in is ecological and environmental harm. If you do a trial on a severely threatened animal, you cannot just use large numbers because you might be threatening the very existence of that animal. So this needs to be done with very careful consideration. Then knowledge interest. If this is such that there is a general benefit, then it obviously would score highly, whilst if it's some negligible problem, then we would not consider it. And then economic interest, there is no doubt, although one would think, right, well, this doesn't play a role in these ethical considerations. They certainly do, because economic benefits can be economic benefits to human well-being and being able to function, etc., etc. So the economics just simply do not disappear from what we have here. So then we've got this funny little calculation here. Calculate the total interest of the ultimate goal. And this is now this IUG, which is these different formulas. I'm not even going to try to name them, but if we just go one page back, please, Biosha. So you can see there that's human interest is the first H, knowledge interest is K, and economic interest is E. If we go to the next page again, and you can see these are the various formulas that we sort of try to fiddle and try to get some idea of a combination of the weighing of these factors, which we then call the IUG. And funnily enough, we put them into these various formulas, and then we say, right, well, we'll take the highest. So this, again, sounds dodgy. It sounds subjective. But in fact, it does, if one looks at this in total, it does get you closer to not just simply a decision of emotion. So let's go on. Then humaneness and relevance. These factors are very important. Replacement possibilities. If you're doing an experiment on feeding trials in chickens, then there's no replacement possibility. Then you're going to be using chickens. So as opposed to that, do you want to use chimpanzees? Are there other alternatives? Then we can say, right, well, we'll rather use mice. So these are important factors and weighing factors that we need to take into consideration. General method soundness. This is where a scientist needs to state his case clearly. There needs to be a proper scientific motivation. Humaneness. I write here the 3 R's. The R's are replacement, refinement, and reduction. So we want those factors to weigh. Least refined is 0, most refined is a 10. Then necessity or relevance. You can say, well, whatever you're trying to research here is totally irrelevant, so why should we allow animals to be suffering because of that? If it's cancer research, it's highly relevant, so then it has a much greater score. The probability of success, this sounds weird, but there is in the literature normally a body of information which would give support for your using an animal model or using animals in a trial, and that would allow us to assess whether this has got half a chance of success. If the chances are completely remote, then that doesn't justify the use of the animals. The quality of the research application, again, you can see we need to have a decent research application, otherwise this doesn't hold. And these factors are all added up, and then we divide this by 60, and that would then give us a fraction. And if that fraction, that R fraction, is less than 0.65, we already reject the proposal. We haven't even looked at any of the other factors. But if this doesn't get to 0.65, it tells us, well, the application is weak. And you can see there are a whole lot of factors here, and they've been weighed up in tens and weighed up against each other. And this does get to a meaningful answer, even if it looks a little bit funny. And then we take, in actual fact, human interest as this IUG factor that we calculated at the beginning times R, and that gives us this HI. And as I say, if the R, this previous factor, is less than 0.65, we reject the proposal. We haven't even looked at the animal side yet. So these are the benefits for humanity and weighing up various things from the human side. So now let's get to the other side. We start assessing the harm to animals, the actual discomfort, where we have a scale of 0 to 4. Some procedures can be exceptionally painful, and painful is one, but then the length of the discomfort, as indicated in the second factor, the duration of the discomfort, that is an important factor. So we do not want to have the animal suffering for a long time. And then also what is important is, should there be pain and suffering and over a period of time, there needs to be proper use of medication, of painkillers, etc. And that's where our veterinarians in the faculties come in, and they will supply that support service should it be required. So very important from the animal side. Then the number of animals to be killed. This is funny. It's again placed into this category of 0 to 2, where 0 is less than 10. A 1 is between 1 and 100, and a 2 is greater than 100. It sounds bizarre because it almost looks like a logarithmic scale, but it is. And then total discomfort, which is this scale that's now used here for all of these factors together. And it gives this total discomfort measurement. And if we can go on to the next page, please. We also do a number of other considerations, the psychological complexity of animals. Humans we always view humans to be the most important, and we say that we can perceive all sorts of psychological things around us and so on. And relative to us, we have this psychological complexity assessment. And these are then species considerations. So non-human primates have a 1, other vertebrates have a 0.5, and cold-blooded animals have a -0.2. This has undergone quite critical evaluation in recent years, in that we now consider animals such as octopus to be highly sentient. Yes, they are cold-blooded, but they are certainly capable of assessing and sensing all sorts of things. So this is not something that stands still. Sociability scores. Many animals are gregarious. In other words, they live in herds. And if they are taken out of the herd context, if they're kept singularly, then it means that they stress a lot, unless they are housed in such a way that they actually have contact with adjacent individuals. Sheep, for example, are always in a herd. And if you want to use them in a separate feeding trial, you create a situation whereby they can be visually in contact with each other to reduce the stress that's induced by that. So then these factors are again assessed together to give you a total factor, and then the previous T plus P plus 2, so-called additional weighting, gives you this animal interest weighting. And if we go on to the next page, if the human interest is greater than the animal interest, then the experiment is admissible. And if less, then the experiment is not admissible. So you can see this is a sort of an iffy thing, is this now so cut and dried? Is it so easy? And it's not. Do we do this with every application? No, it's not necessary. Because in some applications, if we look at a feeding trial or something, the amount of suffering is minimal, the amount of interaction between the animals is permissible, all sorts of regulations are met, and then we do not need to go to the depths of doing this. However, You can use a spreadsheet, and I've actually developed a little spreadsheet with which you can fiddle with this. It would mean that we would have to go out of this program to look at the spreadsheet, so we can do that a little bit later should there be interest in this. But it does allow you then to calculate this should you want to for every trial, and you can do it on your own. We can circulate this to anybody that might wish to use it. But it's not necessary in all applications, but in complicated ones, it certainly is. A number of years ago, if we can just perhaps go to some examples, let's go to the next page. Examples, COVID and HIV vaccine trials. COVID obviously was a hot topic 2 years ago. We start forgetting about COVID, but it certainly is still with us. HIV vaccine trials, HIV most probably won't go away. And the question is, do we want a vaccine? Yes, we certainly do, certainly in South Africa. You can also see that in the case of an HIV vaccine, it's of importance to South Africa. Other countries might not want to see that it's so important that there is a vaccine trial, because it might mean that HIV doesn't occur in those countries, or that they use antiretrovirals and so on. So these measurements are also by country, so to speak. How would the acceptability change with the type of animal used? Dramatically. As I explained earlier, if you were to use higher primates for trials, this questions the ethics and the ethical use of those animals severely. And in modern ethics applications, we actually do not consider the use of higher primates to be a positive thing at all. Higher primates have got all sorts of interactions, social interactions, and also they are threatened. So there is a conservation value to keeping these animals in nature rather than keeping them in research institutions. So these are all factors that need to be weighed up. So if we go to the next slide, let's look at an animal feeds trial. How important is this economically? Definitely yes, because we are dependent on more and more nutrition for the human population. So those are strong motivations for such a trial. Which type of animal? Again, single stomach animal like chickens, ruminants like cows, hindgut fermenters like rabbits. These are all considerations. How many animals do you need? Are the animals justified? Yes, they are, because we need to feed the human population. And then there are factors which play a role here, is how much methane is the trial producing? Cattle, through digesting food, produce methane. And methane is a gas which leads to global warming. And this is one of the factors that counts against cattle production in general in the world. So these are factors that we look at. Then we look at the wildlife sampling projects. What are the benefits for the species? In South Africa, we've got tuberculosis, which has crept into the buffalo populations in Kruger Park. And we need to assess this and to be able to try to assess whether there are ways in which we can reduce this. And what happens is that the TV bubbles over to the predator species like lions, and this has a big effect. So these are all factors that have to be weighed up. Just one back, please. There are important considerations in wildlife sampling. Inbreeding in populations, like the cheetah has gone through a bottleneck, in other words, has gone through an inbreeding problem. And in that instance, it's important to assess this, but again, it has to then be done. The sampling has to be done non-destructively, which can be done, but it needs to be done with care and with the necessary regulations and permits in place. So, right, but let's get to the last one. So the harm-benefit analysis, the harm-benefit assessment is a handy tool for guiding us to approve or not approve projects. And And what I want to bring across is that many of the criteria are scientific and they're not based on perception. They therefore try to be more objective and less subjective, which is difficult because animal ethics is something which involves emotion, so it cannot be entirely objective. But certainly, in order to justify animal experiments, they need to be properly motivated. And as indicated on the application form, you would see the complexities. And this cannot be taken lightly. We need to treat animals with due respect, and that is what is expected of us at the university. So thank you very much, and I can take any questions should there be any. It may be that you are a PhD student and that you are considering using animals. Many PhD students or many students are actually asked to do their ethics applications themselves. Here is the animal ethics spreadsheet, which one can fiddle and fill in values. What I've done here is I've just filled in a couple of values. On the left is the benefit, and on the right is the animal harm. And there is this calculate the interest of the total goal. You can see that I get to the highest figure there of 11.5. Then there's humaneness and relevance, that R factor, and you can see if you feed in a number of factors, if there's no replacement possibility, and then you put in all the other values, you get to a value in this instance of 0.6667. It's greater than 0.65, so you could say, right, well, this is justified. Multiply the two with each other, and you get a figure of 7.66. On the right-hand side, we then look at the animal side, the discomfort, the duration of the discomfort, the number of animals. We feed that into a formula, and we get a figure of 3. Then we have a psychological complexity of the animals. In this instance, let's say they are chickens or therefore other vertebrates, giving you a 0.5 there. Sociability scores, highly gregarious. Chickens are gregarious. We feed those together, we get 1.5. And then if we do this total animal interest, we get 6.5. And we look at the total human interest, and we get to 7.6, as opposed to the animal interest, which is 6.5. And we say that the human interest is therefore greater than the animal interest, and therefore that means that the trial is justified and may go ahead. You can see it looks a little bit simple, but it is a tool to guide us. So I see there is a hand up. If you could unmute yourself, then I can hear you.
Hi, Prof. It's Anel Sadi. I don't know if you still remember me.
Oh, yes, I do. I know there are very many of my students that are still in positions at Tygerberg, and I look back on those quite proudly, in fact. Yeah.
Thanks. So what I don't see in this spreadsheet is any consideration of the relevance of the choice of model. So you can lower your score by going for other vertebrates, thereby species consideration, but it might not be an applicable species, like this whole move away from rodents to zebrafish, which I don't actually, as a scientist, agree with. But it gives you a better score, and then therefore supposedly a better chance of getting your ethics approved.
Yeah, but I can assure you that the experts on the committee, at this point, if you were to present a mouse model for cancer as opposed to one for fish, and I know my way through fish because I've also done a huge amount of fish work in my own private capacity. Fish are just phylogenetically far removed from mice, and in that instance, certainly under my chairmanship, we will not move towards zebrafish for everything, because zebrafish have got their place, certainly, but in terms of modern closer to humans, I'm not allowed to speak out of the committee in terms of actual applications that we see, but we obviously see quite a number of applications for rodents which have got, which simulate the human condition. And there is no way that zebrafish are going to replace those, because they're refined models, they're highly specific. I mean, an example of cancers of various tissues or diabetes or something like that, you're not going to replace a fish, at least a mammal, with a fish. That is just not on. So although the model, this figure here would, account against it, in a model, I mean, I don't think that this thing has been specifically developed for a model. And we obviously often deal with models. But I'm not concerned that we're moving away from rodent models.
Okay, thanks.
So,
can I ask another question?
yes, sure.
On a totally different topic. So
Yes.
I'm completely with you on not using or being unwilling to use higher primates for testing, or for animal experimentation. But didn't it used to be the case, or at least the sort of the accepted wisdom in the past, that you needed to do higher primate studies before you could move into human trials? Are we skipping that step now, or where is that going?
So in terms of your scientific argument, you are 100% correct. You would want to move to an animal that's closer to humans before you could come to a definitive answer. And I'm fully in agreement on that. However, the just simple, well, there are factors that actually weigh against the primate model, which don't necessarily have so much to do with ethics, but have to do with cost. And that is that keeping primates in facilities is so expensive that you can hardly afford it. So, there are a whole lot of things that count against using primates. I know, for example, that if one goes back in the history and you have a look at heart transplants in South Africa, which Christian Barnard was the one who actually was the pioneer of those, developing those techniques. And the reason why he took the step of going towards humans was because of the baboon experiments that were done in South Africa. And there it's just about impossible to replace another animal with anything but a primate. So these things have to be very, very carefully assessed, and we will not categorically sweep something off the table for a species reason. All of these things have to be taken into consideration. There are trials where I can maybe mention an instance. A behavioral trial a number of years ago was done on mice, in which there was a trial in which they created a mice city. So they had all sorts of buildings and places where the mice could live, not standard sort of mouse housing, but a huge area. And then they let mice run free and multiply, and they fed them ad lib or something like that. And then they noted how the population expanded and how the mice started becoming very competitive with each other. And they made deductions about that. That was going to be how humans would behave if they were under higher densities. And that this was mentioned in the literature recently. These trials were done about 40, 50 years ago. They actually influenced US Senate meetings on human populations. And now those experiments are completely dismissed. We do not accept rodent behavior as an acceptable comparison to human behavior. So there are things that have changed, certainly also with regard to, primate use. But there's some things that you can replace, but there are others that you can't. And we won't categorically say, no, we can't deal with this. Obviously, Stellenbosch University has got certain facilities, and the requirements that we have to meet are those that are laid down in SANS, in the national standard. And those, when we assess a trial with regard to how the mice are kept or the rats are kept, that always has to be according to those SANS standards. And I must be quite honest, I haven't looked at the SANS standards for primates, but I could imagine that they would be pretty stringent.
Okay, thank you.
What I can perhaps just add is, in some instances, the actual presentation of an application may be somewhat difficult, and we've found that some researchers battle to formulate them. And we've actually gone over to a system where, if there are problems, and also if we have an application that we recognize as having shortcomings, but where there might be misunderstandings, we in actual fact organize a meeting with the applicant and solve the problem by just speaking to them and telling them what we require, instead of it being this black box. That's one of the problems of the online application, that you can't sort of be asked or ask somebody and be reassured. But that's why Biosha and Winston are there. They can also answer these questions. The idea is that we want to facilitate research. We don't want to be a burden. Any more questions?
If there are no more questions, I would just like to thank you, Prof, for your presentation and for providing the harm benefit calculator spreadsheet. It will be very, very useful for all animal researchers in the current meeting and moving forward, for those that require a consultation, that require perhaps a way to address the research using a calculation where they can assess human interest versus animal interest. And colleagues, for those that are present, I would be happy to share Prof's calculator spreadsheet. You can just let me know via email or in the chat, and I'd be happy to do so after the presentation.
Thank you, Biosha, and thank you everybody for attending.